Imagine if you could edit or snip out hereditary diseases like cancer from your gene sequence. Incredible, right? If anything gets attention here at the World Economic Forum in Davos, Switzerland, it's breakthroughs like that co-developed by UC Berkeley biochemist Jennifer Doudna. And you're here with us, having just done a panel this week with Joe Biden, the Vice President of the United States, called Moonshot to Cancer Cures. Could this really be that?
I think it's a very exciting time in science. I think one of the things that this panel highlighted was the collection of technologies that are coming together right now to fight diseases like cancer. And I'm delighted that our administration is taking the lead on this.
Well, they are, but yours in particular, the CRISPR-Cas9. Explain how this would work.
This is a technology that came about through an international collaboration with a colleague of mine, Emmanuel Charpentier, and our students and postdocs. And what we figured out was that we could harness a bacterial immune system as a genome editing technology, a way to precisely change the DNA sequence in cells for therapeutics and other applications.
Well, as I understand it, it searches through billions of cells to target what? Maybe a hereditary gene that would be a negative, like the BRCA cancer gene?
That's correct. It's a system that can be literally programmed by scientists to recognize a particular DNA sequence and make a change at that place in the genome.
You know, I was fascinated to see that initially this type of thought was being used to change the pattern on butterfly wings or maybe modify plants.
Yeah.
There would be huge applications certainly in agriculture, correct?
Indeed, absolutely, and that's already happening.
Mm-hmm. But it's really the focus on the human life, I think, that people are interested in, but there's controversy about this, right? How do you make sure that it is used correctly and not to edit out certain features and things that are the choice of humans versus the choice of nature?
Well, like with any technology, there are applications that I think, cause us to pause. And I've been an advocate of having an open global discussion about this technology and how we should use it responsibly. I should add, though, that I think right now one thing that holds us back from doing what you just said is that we don't know enough about our own genome to be able to make changes that would really change us as humans quite yet.
It hasn't been tested on humans, correct? But there is a Chinese research team that was testing it on human tissue?
Human embryos, yes.
Human embryos.
Yes, right. They did experiments in non-viable embryos. Mm-hmm. And what their experiments showed really was that the technology, although it works in those cells, is not quite ready for that kind of application.
You're a scientist, so you don't work on human subjects, but what is your dream for this technology that you've co-created?
I'm so excited about using this to really improve human life. I think that the potential to do things like remove genetic disease from humans and even to help us treat diseases like cancer is really very tantalizing. And frankly, I think it's not too many years away.
Professor, I'm trying to do this moonshot thought here. Could it scan billions of cells, find, say, Alzheimer's or Parkinson's in a fetus, and then be able to edit that out one day?
Well, I think that's really a possibility with this. And again, my feeling is that we don't want to go there yet for clinical application until we've really shown the technology is safe.
How precise is it so far?
It's pretty precise. I think what people have shown in when they work in whole animals or in what are called primary cells is that it's really a very accurate system for making changes.
What have you been able to alter or edit or snip in animals?
Oh, so many things. Lots of applications. I think, let me think of a couple of examples. I mean, people are using it to make better animal models of human disease, like lung cancer. Mm-hmm. People are using it to create models that are showing us how we could use this technology to one day cure Duchenne muscular dystrophy. 3 papers that just came out about that topic. So I think the potential is very tangible.
Wow, it's exciting, and it's so exciting that people are talking about maybe a Nobel Prize for you and your team. How much do you think about something like that?
Very little.
Yeah.
I'm a scientist. I focus on the science. I'm really excited about advancing the technology and doing what I can to help it get into the real global use, I guess.
Well, in these cases, a lot of people pounce and say, well, I was part of this. There's a little bit of a patent dispute right now?
Indeed, there is.
How do you see that playing out?
Well, it'll probably take a while to play out, and again, I stay focused on the science and what I do best.
Women in STEM, science, technology, math, engineering, do we do something wrong in the way that we're encouraging young ladies to get involved in that kind of study?
I think it's hard for women still, unfortunately. There's still a lot of unintentional bias that I detect. And I think for girls, it's hard for them sometimes to feel that they have the ability to go forward.
Well, what inspired you when you were a kid?
I grew up in Hawaii in a rural town. I've always loved figuring things out. I had great encouragement from my dad, especially, who was not a scientist. Nobody in my family was a scientist. And I just, I think I have a curiosity about me, that just a sort of a passion that I can't burn it out of myself.
Well, it brought you here to Davos, and maybe it'll bring you a Nobel Prize. We wish you good luck.
Thank you very much.
Thank you so much.
Real pleasure to be on the air.
Jennifer Doudna.